Gel electrophoresis is a fundamental technique in molecular biology used to separate DNA fragments based on size. In this video, we'll demonstrate how to prepare and run an Agarose gel. We need the following equipment from your student kit. A gel box. It holds the gel in the running buffer. It has two electrodes, a negative cathode and a positive anode. A casting tray, which shapes the gel and holds the wells for loading samples. A double-sided comb creates wells in the gel where DNA samples will be loaded. Once the gel solidifies, the comb is carefully removed to leave evenly spaced wells. Two baffles which help the gel stay in place while it's cooling, and the lid which has two power source cables. In some instances, you will need to prepare the Agarose gel. First, weigh the correct amount of Agarose powder and mix it with TAE in a flask. Gently mix and allow the Agarose to hydrate for a minute. After hydrating the Agarose, heat the solution in the microwave, mixing occasionally to ensure complete dissolution. Avoid overboiling, as this can cause buffer evaporation, altering the gel's final Agarose concentration. While waiting to pour the gel, place it in a suitable water bath to maintain its temperature. Before assembling the gel rig, ensure the gel box is clean. Place the casting tray in the center of the gel box. Insert the baffles on both sides of the gel box into the ridge, with the smooth side facing inward toward the gel. Push down on the baffles to ensure they are secure. The comb has two sides, one for smaller wells and one for larger wells. Check with your instructional team to determine the appropriate well size for your experiment. To set it up, loosen both knobs and slide the comb into position, adjusting the height so it sits just above the gel tray surface without touching the bottom. Make sure the comb is level before tightening the knobs to secure it in place. Since DNA migrates from the negative to the positive electrode, place the comb closer to the negative electrode to ensure proper migration. Leave a small gap between the comb and the baffle. With the rig set up, it's time to pour the Agarose gel. Retrieve the Agarose solution from the water bath and check that CyberSafe has been added. If not, mix it in now to allow for DNA visualization. The solution should have a slightly peach color. Gently pour the Agarose gel into the casting tray, ensuring an even distribution. Let it sit undisturbed until it solidifies, which should take approximately 20 minutes. There are multiple ways to prepare your DNA sample. If using a parafilm sheet, clearly label the sample before adding the appropriate amount of loading dye and mixing directly on the sheet. Alternatively, you can mix the DNA and loading dye in a labeled 1.5 milliliter Eppendorf tube. Once the gel has solidified, gently remove the comb and baffles, taking care not to disturb the gel. Measure the required volume of TAE buffer and slowly pour it over the gel, ensuring the wells are completely flooded and the gel is fully covered. Carefully load each sample into a well using a micropipette. Position the tip just above the well, gently depress the plunger, and allow the sample to flow smoothly into the well. Some common errors to avoid are bubbles in the pipette tip. If you see bubbles in the tip, redraw the sample to avoid dispersion in the well. Also, keep the pipette tip away from the bottom of the well to prevent sample loss and tearing of the gel. Place the lid on the chamber and connect the electrodes, black to black, cathode, negative, and red to red, anode positive. Then turn on the power supply, set the voltage and run time, and press Run. When the gel run is complete, turn off the power supply and disconnect the electrodes. Remove the lid and gently lift the gel casting tray. To drain excess liquid, place a finger on the edge and tilt the tray. Carefully transfer the gel to the box by sliding it from the casting tray. Now you're ready to observe your gel. Cover the blue LED trans-illuminator and turn it on. View and photograph your gel through the top of the enclosure. To clean up, dispose of the gel in the biohazard bin. Rinse the rig and its components with water, dry them, and return the rig to your student kit.